to some bones, some tiny bones, malleus, incus, and stapes. Malleus, incus, and stapes. So, as you know, with these small bones, a human has 206 bones. Each individual has 206 bones. If we calculate these bones in this regard, so we have three little bones in our body in the middle ear, and these are malleus, incus, and stapes. A small window in our inner ear. So after middle ear, we have inner ear, and as you can see, stapes is connected to a window which is called oval window. Oval window, because of its shape, it's called oval window. And as you can see, we have a round window which is located lower with respect to the oval window. In the inner ear, we have cochlea. Cochlea, which is looked in Persian we call cochlea, which is similar to a snail. Okay, and we have some semicircular channels, semicircular canals. Okay, and oval window, round window, and cochlea, and finally this is the nerve, acoustic nerve, acoustic nerve, or vestibulocochlear nerve, vestibulocochlear nerve, or acoustic nerve. The main you learn about the function of this part of the inner middle, which is about the sense of equilibrium. Equilibrium. But the function of the cochlea is in terms of the hearing. So the inner ear is involved in sense of equilibrium and at the same time in the hearing. Okay. Conduction of sound from the tympanic membrane to the cochlea. Figure 53.1, the previous figure that you saw, shows the tympanic membrane, commonly called eardrum, and ossicles, small bones, ossicles, which conduct sound from the tympanic membrane through the middle ear to the cochlea. Cochlea is located in the inner ear, and tympanic membrane or eardrum, which is connected to a small bones which is called ossicles, are located in the middle ear. Okay, attached to the tympanic membrane is the handle of the malleus. The malleus is bound to the incus by minute ligament. So whenever the malleus moves, the incus moves with it. The opposite end of the incus articulates with the stem of the stapes, and the faceplate of the stapes lies against the membranous labyrinth of the cochlea in the opening of the oval window. You learn this topic. This is eardrum or tympanic membrane, which is connected to the malleus. Malleus is connected to the incus. Incus is connected to the stapes. Stapes is in touch with the opening of the oval window, and sound will be encounter to the tympanic membrane and moves these structures, ossicles, small bones, and finally the vibration will be projected to the opening of the oval window and finally moves the fluid which is filled the inside of the cochlea. Inside of the cochlea is filled with a fluid that we will discuss about this in the rest of the session. The ossicles of the middle ear are suspended by ligaments in such a way that the combined malleus and incus act as a single lever, having its fulcrum approximately at the border of the tympanic membrane. So one action of this ossicle is just like a lever, okay? This is another picture from Fox and Rompolsky that represent the structure of the ear, external ear, middle ear, inner ear. You can see this is tympanic membrane or eardrum, ossicles, small bones, malleus, incus, and stapes, and semicircular canals, cochlea, vestibulocochlear nerve, oval window, round window. Another picture from the Bernal Levy. In this picture, you can see a medial view of the middle ear, tympanic membrane, ossicles, oval window. This is a stapes. This is the opening of the oval window. This is round window. This muscle is important, tensor tympani muscle, tensor tympani muscle, okay? Everyone, please write the name of this window, which is located here. What is the name of this window? Oval window. Very good, Fatima. Thank you so much. As you can see, oval window is the opening of this window, and this window is connected to the cochlea. Cochlea is just like a snail, okay? And in this picture is try to illustrate the shape of this cochlea, which is almost a bit tied up, and you can see that here we have a canal which is connected to this canal that finally leads to the opening of round window, and here is filled with a liquid system, okay, which will be discussed in the rest of this lecture. One important function of the ossicular system in our ears, the small, the ossicular system, the small bones in our middle ear. One important function of the ossicular system is impedance matching. Impedance matching. The amplitude of the movement of the stapes faceplate with each sound vibration is only three-fourths as much as the amplitude of handle of the malleus. The system actually reduces the distance but increases the force of movement about 1.3 times. In addition, the surface area of the tympanic membrane is 55 square millimeters, whereas the surface area of the stapes average 3.2 square millimeters. This 15-fold difference, 15-fold difference from the 55 square millimeters divided by 3.2 square millimeters. 55 square millimeters is the surface area of the tympanic membrane, eardrum or tympanic membrane, and this 3.2 square millimeters is the surface area of the stapes. Stapes is the third osticle which is connected to the oval window. So we have a 17-fold difference times that multiplied by the 1.3-fold ratio of the lever. The levers, these levers, as I told you, these ossicles act as an lever system. So 17 multiplied by 1.3 equals 22. So this 17-fold difference times the 1.3-fold ratio of the lever system causes about 22 times as much total force to be exerted on the fluid of the cochlea as is exerted by the sound waves against the tympanic membrane. So these systems together increases the strength of system about 22 times more and stronger. Okay, this is the main function of the system. Attenuation of sound by contraction of the tensor tympani and stapedius muscle. When loud sounds are transmitted through the articular system and then into the CNS, a reflex occurs after 40 to 80 milliseconds to cause contraction of the stapedius muscle and the tensor tympani muscle. This is an supportive mechanisms to protect our ear, to protect our ear. This is tensor tympani muscle. Tensor tympani. Its function is attenuation of sound when we face too loud, very loud sounds in the environment. This attenuation reflex can reduce the intensity of lower frequency sound transmission by 30 to 40 decibel, which is about the same difference as between a loud voice and a whisper. Regarding the cochlea, functional anatomy of the cochlea. Cochlea consists of three tubes coiled side by side. So, cochlea, which is look like a snail, is composed of three tubes coiled side by side. The first one is the scala vestibuli, the middle one is the scala media, and the third one is the scala tympani. Scala vestibuli, scala media, scala tympani. The scala vestibuli and scala media are separated from each other by Reissner's membrane, also called vestibular membrane. So we have three tubes inside cochlea which are located side by side. The first one, scala vestibula, the middle one, vestibula media, and the third one, the scala tympani. Scala vestibuli, scala media, scala tympani. Okay? This this membrane, this membrane between the scala vestibuli, the upper one is scala vestibuli, the lower one is scala media, and this membrane is called vestibular membrane or Reissner membrane. The scala tympani is the lower one, and scala media are separated from each other by the basilar or basilar membrane. Basilar or basilar membrane. Okay? On the surface of the basilar membrane lies the organ of Corti, which contains a series of electromechanically sensitive cells or hair cells. They are the receptive end organs that generate nerve impulses in response to sound vibration. So this slide is important and simply describes the structure of the cochlea. Three tubes which are located, which coiled side by side, and two membranes between these Reissner membrane between scala vestibula and scala media, and basilar membrane between the scala media and scala tympani. And on the surface of the basilar membrane, which is separated the scala media and scala tympani, we have organ of Corti. Organ of Corti has some hair cells, and these hair cells are the receptive end organ that generate nerve impulses in responses to sound vibration. Okay, this is the first tube, scala vestibuli. This is the middle tube, scala media, and this is scala tympani. So this is Reissner membrane, and this is basilar membrane. And here we have organ of Corti. Organ of Corti. Again, in this picture, you can see that this is clear, and this is the plane of this picture. This is our plane. We have a cross-sectional view in this picture. So this is the scala tympani, scala vestibuli, scala media, membrane, and organ of Corti. Hair cells are located here on the top of the organ of Corti. Again, you can see a scala vestibuli, which is located above the other tubes, scala media, scala tympani. What is the name of this membrane? Please write it down. What is the name of this membrane? Jana, write the name here. Rashid, Mr. Shoman, Ali, are you awake or you slept? Vestibular membrane. Or the other name, Reissner's membrane. Very good, thank you. And here, scala tympani. This is basilar membrane, and this is organ of Corti, and these purple these purple parts are hair cells, and you can see the tiny hairs here. You can see tiny hairs here. This part of organ of Corti, this part called tectorial membrane, and as you can see, these are hair cells, and these are the hairs. Hairs are projected from hair cells to the tectorial membrane. And one important topic is that. Please learn it right here. That scala vestibuli and scala tympani are filled with a liquid which is called prelymph. So we have prelymph in scala vestibuli and scala tympani. But it's a different matter in terms of the scala media. As you can see, scala media is filled with endolymph, or in Persian endolymph. Okay, so the structure of a liquid system which is filled, the scala media is different with respect to the scala vestibuli and scala tympani. Prelymph, here endolymph. Reissner's membrane maintains a special kind of fluid in the scala media that is required for normal function of the sound receptive hair cells, that you know that this fluid is endolymph. Endolymph is filled the scala media. Now, in this slide, you can see a cochlea that is open. That is open. Okay. Fact, we suppose that you unscrew the spring of the cochlea, As you know, cochlea is like a snail, okay, resembles a snail. And suppose that you unscrew the spring of a snail, and you have a lying cochlea, which is consists of three tubes located side by side. The first one, the upper one, is the scala vestibuli, and the middle we have the scala media, and at the lower part we have the scala tympani, scala vestibuli, scala media, and scala tympani. And as you can see, this is the basilar membrane. Basilar membrane. Why we cannot see the Reissner membrane? Why we cannot see the Reissner membrane here? Because in terms of movement of fluid in the cochlea after encounter with the sound vibration, you can imagine that Reissner that you can imagine that there is no Reissner membrane in terms of movement of fluid in the cochlea. So this is basilar membrane, which is important. Reissner membrane function is to keep the endolymph inside the scala media. But Reissner membrane is not important in terms of movement of fluid in the cochlea. So in this picture, for the sake of the simplicity, for the sake of simplicity, we remove the Reissner membrane. And this is basilar membrane, which is important in terms of movement of fluids, okay, in the cochlea. The basilar membrane is a fibrous membrane that separates the scala media from the scala tympani. And now you know that this is oval window and this is a stapes ossicle, which is connected to the malleus and incus, and which in turn are connected to the eardrum or tympanic membrane. Eardrum is here, and these systems transfer the vibration of the sound to this fluid system inside of the cochlea, and we have a movement of the fluid which is filled inside of the cochlea because of the vibration of the sound. So in fact we translate vibration of sound into the movement of fluid inside of the cochlea. This is another picture from Baron and Bullpup. Baron and Bullpup is an important medical physiology textbook which is taught in the highly prestigious university all over the world. For example, in the US, in Harvard University, in Stanford University, Johns Hopkins University, okay? Along with the Guyton and Hall, Baron and Bullpup is a very and highly prestigious medical textbook in with regard to physiology, medical physiology. Scala vestibuli, scala media, scala tympani, Reissner membrane, basilar membrane, organ of Corti, hair cells, hair cells. As you can see, we have two main types of hair cells: inner hair cells, outer hair cells. outer hair cells are more than inner hair cells. For example, we have one inner hair cell compared to the three outer hair cells. But inner hair cells are most important in terms of sense of hearing. And as you can see, these tiny hairs above the hair cell which are projected into the tectorial membrane. This cavity, this tube is with endolymph, but these tubes are filled with prelymph. Basilar membrane and resonance in the cochlea. The basilar membrane contains 20,000 to 3,000 stiff, elastic, reed-like basilar fibers that project from the pancreas, the modulus toward the outer wall, where the distal ends are embedded in the loose basilar membrane. The length of basilar fibers increasing from a length of about 0.04 millimeters near the oval and round window to half millimeter at the tip of the cochlea, the helicotrema. So the tip of the cochlea is called helicotrema, a 12-fold increase of the length of the basilar fibers are exist between the tip and the base of the basilar membrane. This is the base of the basilar membrane. This is the tip of the basilar membrane. As you can see, the tip of the basilar membrane is called helicotrema. Helicotrema. And the length and the thickness of these basilar fibers, which are like the reed, these are reed-like fibers. The length, the thickness, the diameter of these reed-like basilar fibers are different, and they will be here. We have the smaller and thicker one, and here we have the longest and thinner one of these reed-like. So these basilar fibers react to different frequency of sound. These basilar fibers react to different frequencies of sound. The diameter of these fibers decreases from the oval window to helicoidal, so their overall stiffness decreases from decreases more than 100-fold. As a result, the stiff, short fibers, this is important, please pay attention. The stiff, short fibers near the oval window, the color vibrate best at high frequency, whereas the long, limber fibers near the tip of the cochlea, near the helicotrema, vibrate, vibrate best at a low frequency, at low frequency. So this part of the basilar membrane vibrate best vibrates best at every high frequency because these basilar fibers, which are located in the first part of the basilar membrane, are shorter and thicker compared to these one, which are longer and thinner. So these basilar fibers vibrate best at a low frequency. So this part of the basilar membrane react better to low frequencies, and the base of the basilar membrane vibrate best at a very high frequency. So this is one an important way that we can differentiate between the quality of the sound. This is one and very good and the most important way that we can differentiate between the sounds. Transmission of sound waves in the cochlea or traveling wave. Traveling wave. When the foot of the steps move inward against the oval window, the round window must bulge outward because the cochlea is bounded on all sides by bony walls. The initial effect of a sound wave entering at the oval window is to cause the basilar membrane at the base of cochlea to bend in the direction of the round window. However, the elastic tension that is built up in the basilar fibers as they bend toward the round window initiates a fluid wave that travels along the basilar membrane toward the helicotrema. So you can see this is oval window, this is round window. What is this? Please write the name of this section. Yusuf, Zahra, Dua, Noor, Rashid, Jana, Haura, Fatima, Ali, all of you, write the name of this section. Dua, yes, not your name, Dua. What is the name of this part of the cochlea? Hi? What is hi? What is this? What is this? What is this? What is this line? Base. Only base? It's basilar membrane. Very good, Rashid. Rashid, I love you. Rashid, you are the best. Basilar membrane. Very good. And as you can see, this is a traveling wave. Traveling waves. Sound waves reach to this fluid system. This which is filled the inside of the cochlea and create traveling waves on the basilar membrane. And as you can see, different parts of the basilar membrane react best to different frequencies of sound because the basilar fibers which consist these parts of the basilar membrane are shorter and stiffer, so high quality high frequency vibration react acts here, middle frequency waves acts here, and low frequency waves can be represented better at the tip of basilar membrane, also known as helicotrema. As you know, vibration patterns of the basilar membrane from different sound frequency differentiate patterns of transmission for sound waves of different frequencies, and this is a way to differentiate between the sounds. different frequencies, different part of the basilar membrane. Regarding the function of organ of Corti, the organ of Corti is the receptor organ that generates nerve impulses in response to the vibration of the basilar membrane. Note that the organ of Corti lies on the surface of the basilar fibers and basilar membrane. The actual sensory receptors in the organ of Corti are two specialized types of nerve cells called hair cells. You know that. A single row of internal or inner hair cell. As I told you, as you remember, we have two main types of hair cells. Inner hair cells, outer hair cells. So, a single row of internal or inner hair cells numbering about 3,500 and measuring about 12 micrometers in diameter. And we have 3 or 4 rows of external or outer hair cells numbering about 12,000 and having diameters of only about 8 micrometers. You can see scala vestibula, scala tympani, scala media, Reissner membrane, basilar membrane, organ of Corti. This is an inner hair cell and these are outer hair cells. Tectorial membrane, tectorial membrane, tectorial membrane, outer hair cells, inner hair cells, hairs. Hairs projects, hairs which are projected to the tectorial membrane, okay? This picture is from Guyton and Hall. Figure number 53, 6. Basilar membrane and basilar fiber. Here is the scala media, scala media, scala media, scala tympani. Okay? Outer hair cells, inner hair cell. Tectorial membrane, hairs which are projected from the hair cell into the tectorial membrane. Hairs. Okay? And as you can see, inner hair cell is more important compared to the outer hair cell and is connected to the spiral ganglion and cochlear nerve, which finally projects to the brain, to the cortex, auditory cortex, goes to the auditory cortex. Between 90% or 95% of these endings terminate on the inner hair cells, emphasizing their special importance for detection of sound. The hair cell synapse with a network of cochlear nerve endings. Most of these hair cells from inner hair cells creates cochlear nerve endings, emphasizing the special importance of detection of sound with regard to the inner hair cells. The nerve fibers stimulated by hair cells lead to the spiral ganglion of Corti, which lies in the modulus or center of the cochlea. Excitation of the hair cells. Minute hair cells or minute hairs or stereocilia. Minute hairs or stereocilia project upward from the hair cell and either touch or are embedded in the surface gel coating of the tectorial membrane, which lies above the stereocilia in the scala media. So these minute and tiny hairs are called stereocilia. Stereocilia projects to the jelly structure of the tectorial membrane. Tectorial membrane. Bending of the hairs in one direction depolarizes the hair cells, and bending in the opposite direction hyperpolarizes them. This in turn excites auditory nerve fibers synapsing with their bases. So these hair cells movement in one direction depolarizes the hair cells, and in the opposite side hyperpolarizes the hair cell. And as you know, these hair cells are connected to the auditory nerve fibers. So move to one direction, create action potentials which will be transferred in the auditory nerve fibers, and bend in opposite side hyperpolarizes the hair cell, and there are no action potential in the nerve fiber. As I told you, auditory signals are transmitted mainly by the inner hair cells. So inner hair cells are more important compared to the outer hair cells. Hair cell receptor potential and excitation of auditory nerve fiber. Each hair cell has about 100 stereocilia on its apical border. When the basilar fiber bend toward the scala of stimuli, the hair cell depolarize, and in the opposite direction, they hyperpolarize. As you can see, this is a hair cell. This is a hair cell which is zoomed here. This is the hair cell, and these are tiny hairs or stereocilia. These are tiny hairs or stereocilia. As you can see, the tip of these stereocilia is connected together. We have some ion channels, specifically potassium ion channels in the tip of these hairs, in the tip of stereocilia. And these channels are connected together with these link proteins. These or this link protein is very, very small. Even it's hard to be observed using electron microscope. So these connections are very, very small. And as you can see, the tip of these stereocilia is connected together. We have potassium ion channels here, which are connected together. And generally speaking, bending of these stereocilia, for example, bending to the right, into the right, bending to right, opens these channels. Now potassium ion can go inside easily. As a result, we have depolarization in this hair cell. Depolarization leads to opening calcium voltage-gated calcium channels. Now calcium ion go inside, and this calcium ion increases exocytosis of neurotransmitter, and these neurotransmitters are glutamate. And as you know, glutamate is one or most excitatory neurotransmitter in the CNS. So this glutamate is released and affect the afferent neuron, the auditory nerve, and excite the auditory nerve and create action potentials which are transmitted to the central nervous system to the auditory cortex, which are located in the temporal cortex of the brain. So this is the simple explanation of the creation and transmission of action potential excitation of auditory system. Okay? You need to know, Rashid, we will have a break rest at 1100. Okay? At 1100, 2 minutes later, we have a brief break, short break. Rashid, I guess that what do you want to say to me? Yes? What do you want to tell me? Okay. Regarding the endocochlear potential, you need to pay attention that, as you know now, the scala media is filled with endolymph, but scala vestibuli and scala tympani are filled with another liquid, another substance which is called prelymph. So you need to know that endolymph is different in terms of chemical substances, regarding the ingredient substances, regarding the electronegativity compared to the prelymph. And as and you should know that the scala media is filled with fluid called endolymph, whereas the scala vestibula and scala tympani of prelymph, which is almost identical to cerebrospinal fluid (CSF). In the lymph, this is important, in the lymph contains a high concentration of potassium and low concentration of sodium, which is different with the most other parts of our body. And because of this difference between sodium and potassium concentration and electrical potential of about positive 80 millivolts or so called the endocochlear potential is generated by the continual secretion of potassium, the scala media by the stereo vascularis. So because we are the continuous secretion of potassium ion into the scala media, we have a endocochlear potential here, and we have about positive 80 millivolts endocochlear potential here with respect to the scala vestibuli and specifically scala tympani. As I told you, difference between scala media and scala tympani is important because organ of Corti is located between scala tympani and scala media. Okay, we have 3 or 4 minutes short break and I'll be right back ASAP. Hello everyone, come back and we are about to continue the rest of this lecture regarding the sense of hearing, and after that chemical senses. Welcome back. Thank you, Fatima. Welcome you. Okay, this is important that pay attention that as I told you, we have in the cochlear potential about positive 80 millivolts in the endolymph liquid, endolymph fluid, and again, the hair cells have a negative intracellular potential about minus 70 millivolts with respect to the prelymph. So we have minus 150 millivolt with respect to the endolymph at their upper surface. Here is filled with endolymph with positive 80 millivolt electronegativity, and inside of her cells we have about minus 70 millivolts electronegativity. So we have about 150 millivolts electronegativity differentiation between inside of the hair cells and the endolymph, which is a huge difference. Okay, this high electric potential at the tips of the stereocilia synthesizes the cell an extra amount, thereby increasing its ability to respond to the slightest sound. Regarding the central auditory mechanisms, as you can see, we have the cochlea here, which is connected to the auditory nerve. Excuse me for a minute. As you know, acoustic nerve or vestibulocochlear nerve is the eighth cranial nerve and is projected from the middle ear into the auditory cortex, which is located in the temporal part of the brain. As you know, primary auditory cortex is located in the temporal lobe of cortex. So, regarding this pathway from the cochlea, from the inner ear into the primary auditory cortex, you should pay attention that the nerve fibers from the spiral ganglion of cortex